Okay, so this is our video on the development of Danio rerio, which is also known as the zebrafish. This is by myself and my two lab partners, An and Sean. Okay, so I want to begin this by just giving a little bit of background information about the organism of choice. As by the title slide indicated, it is the zebrafish, which is part of the phylum Chordata. This indicates that it has a true nodal cord, pharyngeal gill slits, a dorsal hollow nerve cord, and a post-anal tail. Additionally, its scientific name is Danio rerio. This is a model organism for developmental research with several factors playing into this aspect. One being that it has a transparent embryo and zygote, and you can see all of these stages of development as they occur. It has a fast generation time with approximately 48 hours until a moving viable organism is present. And also it has inexpensive housing, such as you can have 10 organisms in one enclosure, and it doesn't take a lot of space to house them or a lot of money to keep them alive. Lastly, they're used in numerous types of research, as explained by Dooley et al. They're used all the way from kidney disorders to cardiovascular disorders, and used to study the development of other organs such as the eyes and heart. Okay, so before diving into the development of the zebrafish, it's important to know that the type of cleavage that a zebrafish undergoes during its development. So a zebrafish undergoes a meroblastic cleavage, particularly a discoidal cleavage, which is found in fish, reptiles, and birds. And this basically means that it has a dense yolk throughout most of the center, which is indicative of a telolecical style of meroblastic cleavage. And as you can see in the second row of the diagram, that is the style of cleavage that we will see. We will see a yolk, and then we will see the 2-cell stage, the 4-cell stage, and so on throughout the development. Okay, so here are all the developmental stages for a zebrafish, with there being 35 in total. These range all the way from the zygote, which is pre-fertilization, to 48 hours post-fertilization. This diagram with all these subsequent steps were used to help identify which stage our particular zebrafish were in during the developmental process.
This is the zebrafish in the 2-cell stage going to the 4-cell stage. This is approximately 50 minutes to 1 hour post-fertilization. At this point, the individual cells can be clearly observed at the animal pole, while the yolk can be observed at the vegetal pole. Next up is the 4-cell stage. This is approximately 1 hour after fertilization. This stage occurs within minutes of the 2-cell stage, showing just how fast the development of the zebrafish occurs. Ensuing development continues from the 4-cell to the 8-cell stage. This stage is typically observed about an hour and a half post-fertilization. Again, as in the 2 and 4-cell stage, the cells can clearly be observed at the animal pole. Moving further into development of zebrafish, the 128 cell stage was observed. This stage is typically observed a little past the 2-hour mark of development. The aggregation of cells is clearly visible at the animal pole, showing how meroblastic cleavage occurs.
Further into the zebrafish development is the epipolar stages. Shown is a 70% epipolar stage with visible germ ring. The blastoderm envelops 70% of the yolk at around 5.5 hour mark post-fertilization. (90% epipolar stage is shown above. This stage is characterized by the second brain rudiment. At this point, the blastoderm continues to move down around the yolk. The cell then enters the 15th somite stage. By now, the yolk is completely enveloped. After approximately 17 hours post-fertilization, eye primordium and optic plate coat are now visible along with the formation of somates.
Alrighty, as we can see, we are between the 24 and the 28 hour mark. You can see the eyes are starting to finally develop. Alrighty, as we can see, we are between the 24 and the 28 hour mark. You can see the eyes are starting to finally develop, and
It's alive! Oh, it's alive! It's alive! It's alive! It's alive!
finally, we have one happy little fish that is viable, swimming around, and just enjoying its day. On a final note, I would just like to take a moment of silence for the ones that did not make it. In one of the developmental trials for the zebrafish, there was a parasite present that did, in fact, kill quite a few of the developing zebrafish. With that, this concludes our zebrafish developmental video. I would just like to say thank you for watching, and
Bye! Have a beautiful time!